Good afternoon. I'm Ron Bache, President and CEO of Aquesys, and it's an honor and a privilege to be with you here today and share with you some insights about Aquesys. I think if we can go to the first slide, I'd appreciate it. Thank you. So as we've heard, glaucoma is the world's second leading cause of blindness, and it's estimated to be between a $4 and $5 billion market. And in terms of current therapy, medical therapy is very effective, but over time it lacks compliance and efficacy. Today's surgical procedures are highly effective, but they're invasive. So as you heard Dr. Ahmed speak about, there's this large chasm that exists between currently available medical therapy and currently available surgical therapy. Excuse me. And so at Aquesys, our promise is to be able to deliver traditional surgical IOP reduction, but in a minimally invasive fashion. How are we able to do that? So what we do in an ab interno to subconjunctival approach is that we actually utilize the proven outflow mechanism of action that is today's standard of care for glaucoma surgery, the subconjunctival approach. But we've developed a minimally invasive ab interno approach to the subconjunctival space and an implant that is also very unique in its design as well as its material so that it will behave as intended for a long period of time. And those two married together allow us to achieve significant surgical IOP reduction. And thus, I think it's very important that our differentiation is we're minimally invasive and highly effective. A bit about the company. We were founded in 2006 by the Innovation Factory based upon technology that was invented by an MD, PhD at the Lions Eye Institute. We have 30-plus patents that have issued or are pending that cover not only our technology but the surgical glaucoma space. And we're very fortunate to work with many of the leading glaucoma and cataract surgeons around the world in developing our technology, including Dr. Ahmed, who's been with us since the very beginning and helping us innovate, and Dr. Rick Lewis, who is here, who is our medical monitor. And we've been fortunate to secure $50 million in venture capital from top-tier venture capital firms, SV Life Sciences, the Carlyle Group, Acuitive Medical Ventures, Longitude Capital, and Rowe. As we all know, it takes an incredible technology to be successful, but as you heard Bill say, it also takes an incredible team. And I'm humbled and proud that I think we've been able to assemble just a world-class group of individuals that I think are best in class at their disciplines. And so combined with that team and our technology, we're very confident that together with the clinicians, we're going to be able to very much positively change the way glaucoma is going to be treated in the future. A little bit about our technology. We utilize a permanent gelatin implant that's about the size of a human hair that gets implanted from the anterior chamber to the subconjunctival space ab interno. And it has unique characteristics, both in terms of its material and its design, that mitigate or eliminate many of the traditional problems that we have seen in glaucoma and glaucoma implants. The implant comes preloaded in a disposable IOL-like inserter, and it's a dehydrated state. It's stiff. But when it comes in contact with the aqueous humor of the eye, it becomes very soft and very flexible, almost like a wet noodle, if you will, that maintains its structural integrity. So it is very delicate and gentle to the tissue that it surrounds it, and very non-inflammatory. As I mentioned, we use a preloaded disposable IOL-like inserter. I'll show you an example of our procedure, and it's very akin to what glaucoma and cataract surgeons do every day in terms of delivering an IOL into the bag. Our mechanism of action, we're the world's only ab interno to subconjunctival approach, and we think this is very important. I think when we're looking at how you treat glaucoma, mechanism of action is vitally important. And so with our mechanism of action, we use what is currently understood in the subconjunctival approach. We get similar IOP reduction, but we do it minimally invasively. That's really the differentiator. We bypass all of the obstructions, the potential obstructions to outflow in the eye with our approach. So as you heard some of the physicians speak or some of the other members of the MIGS community, there is a debate and emerging science about where is the outflow obstruction. Is it at trabecular meshwork? Is it further down distally? With our approach, it doesn't matter. We bypass all of the potential obstructions for the outflow. And we've been able to show internationally safety and efficacy in early, moderate, and late-stage disease, and I'll show you an example of some of that data. So the advantages is that it's versatile. It can be done with and without cataract surgery with similar effects. It can be done with and without a gonio lens. And so overseas in Europe, where we're CE marked, glaucoma surgeons tend to like to use the gonio lens. Our cataract surgeons tend to find it doesn't aid in their ability to place it well. So it's very versatile in that aspect, very adoptable by a broad range of surgeons. A straightforward and adoptable procedure, I'll show you that. Single implant delivers the desired effectiveness. One implant alone gets us to the efficacy that is right for the patient to get them out of the danger zone and preserve their sight over time. It's removable and or repeatable over the lifetime of the patient's disease, and it's conjunctival sparing, meaning by performing this procedure earlier in the patient's life cycle, you have not taken any other options off of the table for the patient down the road. This is an animation of an aquesynthesis procedure. That's the name of our procedure as termed by our medical advisory board. There is a corneal incision. The inserter is placed to a desired location for the implant, and then it's a simple half a turn of the inserter delivers the implant to the right longitudinal position by itself, both in the anterior chamber and the subconjunctival space. And I'll show you a representation of that with an actual surgical video. So I'd also like to note, with ours, we don't have to hit the canal, we don't have to hit the scleral spur. We have a much bigger working distance, which is why we don't have to have a gonio lens all the time, in that we're just trying to avoid the cornea and avoid the ciliary body. It's a broader area. So what you saw here was the surgeon advance the inserter to the desired location. There's a visualization of the implant in the anterior chamber. And here's going to be a visualization of the implant in the subconjunctiva. And what you'll notice is that's vastly different than you're used to seeing with subconjunctival surgery. That's ab externo. And we'll talk a little bit about that differentiation. How are we able to get to the low IOPs that we are, the highly effective IOPs? Well, we have a great capacity for outflow. That is a fluorescein dye in vivo that was injected into the anterior chamber, went through our Aquesys implant, and demonstrates the drainage capacity that we have. You see both traditional collector channels and it's believed lymphatic vessels are the horizontal branch-like structures that are believed to help aid in the health and maintenance of the long-term drainage of that drainage outflow area. 1 minute of differentiation between a traditional trabeculectomy bleb ab externo developed subconjunctival drainage versus what we do ab interno. And we utilize the term intratenons drainage. I'd like to tell you that I was smart enough to come up with that, but it wasn't me. It was actually Dr. Dick Lindstrom, who's in the room. That's a term that he developed based upon working with us. And so the difference is that with a traditional bleb, you come from outside the eye, you dissect through conjunctiva, you dissect through the tenons, and you break the adhesions of tenons to the sclera. And then you put antifibrotic agents on, and then the fluid flows into that space, and you get more of a raised dome or elevated bleb. With what Aquesys does, we do not dissect tenons, we don't dissect the conjunctiva, and we have minimal tissue trauma. And so there's a graphical representation of our implant placed in the tissue as the architecture is normally found. And thus, the tenons layer subconjunctivally works somewhat like a sponge, if you will. And so we don't get these elevated localized blebs, but we get low diffuse drainage and effectiveness. This is a patient, time course 1 day to 2 years. So you can see pictorially what these patients look like at the slit lamp and gonio lens. A very, very quiet eye. As Dr. Ahmed showed in his picture, it's very hard to distinguish where the implant is in the subconjunctival tissue or in the intratenon space. But yet this patient has an IOP of 8 on 0 meds at 24 months. This is a representation of our international clinical results. It's 118 patients looking at 1 year, 18 months, and 24 months worth of data. Pre-op IOP of 23 millimeters of mercury, median pre-op meds of 3. And what you'll find is that at 12 months to 24 months, we on average are at 14 to 15 millimeters of mercury, and we've reduced meds median by 2. And standard deviation around that IOP is 4 to 5. So IOP reduction from best medicated is roughly 35% at all time points, and we're not seeing a trailing off of effect after 1 year. And I think this is a very big differentiator in terms of when you've looked at other data and our data. These percentage reductions are from best medicated IOP, not washout IOP. So if we were to compute a washout IOP, we're looking at about a 50% reduction if you wanted to do an apples-to-apples comparison. But another way to look at our data is that we're taking patients that come in on at 23 millimeters of mercury, 3 medications, multi-billion-dollar drugs. We're reducing their med load by 2, and we're reducing their pressure an additional 35%. So we think that has a lot of promise in treating that chasm that exists that Ike spoke about. We're getting 7 to 9 millimeters of mercury reduction at those time points, and because means can lie, taking a look at percentage of patients that are at 21, 18, and 16, we're getting 90 to 100% of our patients to 21 millimeters of mercury, or a 20% reduction. Roughly 80 to 90% at 18 to 20% reduction, and 70 to 80% of our patients are at 16 or 30% reduction at those time points. Taking a look at an earlier stage patient population, mean pre-op IOP of 19.5 versus 23, also on 3 meds though, and what do you notice? You notice that we're getting patients in the 13 to 14 category at those time points, standard deviation of 3 around that. Median post-op meds we're reducing by 50 to 100%, and we're getting 27 to 30% reductions from best medicated, which is still from a lower starting best medicated IOP. And then if we look at the per patients, we have 100% of our patients below 21, 80 to 100% of them at 18 to 20% reduction, and 75% of our patients are at 16 or a 30% reduction at these time points. So highly effective in an earlier patient population. And then just to contrast that, we've also been utilized in end-stage disease. So our implants have been used with patients that have failed medications and already have failed a previous trabeculectomy. These patients have 23.4 millimeters of mercury on 2.8 meds at pre-op baseline, and at a year to 2 years, we're at 12 to 13 millimeters of mercury, a 40% reduction, and reducing meds by half or more in this very difficult patient population. So we're having effect early, moderate, and end-stage disease, and we believe that's very much associated with the mechanism of action. And for our implant to be effective, we don't have to hit a specific structure. We just need to make a connection between the anterior chamber and the subconjunctival space. We think that's going to have broad adoption potential. So at Aquesys, we're very confident about our ability to cross that chasm that exists between medical therapy and surgical therapy. We're focused on site-preserving IOP reduction. We think we're able to do that because of our mechanism of action. That is the standard of care around the world. We bypass all of the potential outflow obstructions. We're able to treat broad stages of the disease, early to late, as well as with a broad range of patients. Our mechanism of action is well understood, but we do it minimally invasively. We're CE marked, ready for commercialization outside the United States, and we're currently enrolling our IDE study within the United States. And so we're humble but confident that we're actually able to deliver on the promise of MIGS, which is minimally invasive but also highly effective. Thank you.